Well, hello and welcome you all to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test question paper for the subject Biochemistry conducted in the year 15th June 2023. This is for the subject Biochemistry only. I have discussed only part B questions. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of biochemistry, you can refer to my channel Omni Bio Entrance. Go to the playlist and you will find them there. Then next one. Choose the correct statement from those given below. C5 convertase is formed from C5A and C5B convertase. That is not correct. This is C5 convertase, C5A, 5B. All this happens in your complement system. There are two pathways, that is classical pathway and alternative pathway. There is one another also, lectin pathway. But here they have given only regarding classical and alternative pathway. So C5 convertase is formed from C5A and C5B convertase. That is not correct. It is from C4B, 2B, 3B that C5 convertase is formed, or as in alternative pathway that is C3B, BB and 3B that C5 convertase is formed. And C5 convertase, it will break down C5 into C5A and C5B. So first statement, that is A, is not correct. C5 convertase is formed from C3B and C3 convertase. That is correct. Here if you see C3B and C3 convertase, this one, it is formed from C5 convertase. Here also C3B and C3 C5 convertase is formed. That's correct. The C3A and C3B are formed by C5 convertase. No. C3A and C3B are actually formed by the C3 convertase, not C5 convertase. The membrane attack complex consists of C5A, C6, 7, 8, C8, C9. So here if you see, this is the membrane attack complex. It is having C5B, C6, C7, C8 and poly C9. So D is also correct. C5 convertase is formed from C5AB. That is not correct. So the correct statements here are only B, D. So B and D only. So answer is option 4. Then next one. The following are important steps in plant tissue culture technique which is used to multiply transgenic plants. Choose the correct options. So you have to see for the correct options. Acclimatization of hardened plants. Yes, that's correct. That is after tissue culture, you will take the plant to greenhouse for acclimatization. So A is correct. Spray of insecticide. That is not necessary. Surface sterilization of explant. That's correct. Inoculation of explant on culture medium. Yes. That is also correct. So A, C, D are correct. Okay, you have to find the correct sequence of the steps. Then you will get the answer. So the first one will be surface sterilization of explant. C. Then inoculation of the explant on culture medium. Then the plant will grow there in the culture medium when sufficient because of the suitable environment in the culture medium which is necessary for the plant for its growth. Then you have to take the plant outside for acclimatization. So the answer here is option 1. C, D. Which of the following are types of ELISA? So you have got competitive ELISA, there is no discontinuous ELISA, there is indirect ELISA and sandwich ELISA. So A, C, D. A, C, D only. Answer is option 4. Which of the following are types of electrophoresis? You don't have what is called as competitive electrophoresis, but there is pulse field gel electrophoresis. There is no indirect electrophoresis, but there is two-dimensional gel electrophoresis. So B and D only. Answer is option 2. Then termination of transcription process in prokaryotes is carried out by. So you have got what is called as row dependent and row independent mechanism of transcription in termination in prokaryotes. Ubiquitin pathway, it is for protein degradation, and dicer is involved in miRNA, siRNA like that. So the answer is A and C only. Answer is option 3. Then match the following. So here they have given the disease and the enzyme that is affected. So maple syrup urine disease. Maple syrup urine disease is branched chain alpha keto acid dehydrogenase complex A3. Phenylketonuria phenylalanine hydroxylase, leucine ketogenic amino acid, and valine, it is an essential amino acid. So answer here is option 3. Then next one, IgA, they have given the immunoglobulins and you have to match them. So IgA, it is a secretory it is secretory component. IgA, A4. IgE, it is basophils. Yes, basophils are activated upon IgE. Then IgG, it can cross the placenta, C2, and IgM, it is a pentamer. The structure of IgM is a pentamer. So the answer is option 1, A4, B3, C2, D1. Then puromycin, these are the various antibiotics and the site at which they will bind. So puromycin, it is for a site of the ribosome. Kanamycin is bind to 16S rRNA. Erythromycin, it will bind to 23S rRNA, and streptomycin will be for 30S ribosome. So A2, B3, C4, D1. Answer is option 3. Then next one, actin. Actin, it is a component of thin filament. A4. That is there in option 4 only. So A4, myosin, component of thick filament. Tropomyosin, it binds along the length of thin filaments, and troponin binds along the thin filaments. So the answer here is 4, that is A4, B3, C2, D1. So angiotensin 2. Angiotensin 2, it stimulates thirst. A3. A3 is there only in option 3. Okay. Dopamine. Dopamine is an emotional response. Serotonin induces sleep, and endorphin, it controls body temperature. So the answer here is A3, B2, C4, D1. Then next one, sucrose. Sucrose is a product of photosynthesis. A4. Lactose. Lactose is a major animal energy source. Trehalose. Trehalose is a major circulatory sugar in insects used for energy. And maltose, it is a dimer derived from hydrolysis of starch and glycogen. So the answer here is option 1. A4, B3, C2, D1. Okay, now match the list. The amino acid with maximum number of codon is for serine. Out of these, maximum number of codon is for serine. The characteristic amino acid in lysozyme active site, that is glutamic acid. The amino acid precursor in heme biosynthesis is glycine. The amino acid contributing the most to protein absorbance are tryptophan. D1. So the answer here is A2, B3, C4, D1. So that's it. Thank you for watching my channel. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Biochemistry CUCET entrance exam, you can refer to my channel Omni Bio Entrance. Go to the channel playlist and there you will find the question papers. So thank you for watching. Please do like, share and subscribe.